to introduce today's speaker, Dr. Luciano Moreira. He's a researcher at Fiocruz Foundation in Brazil, where he's been since 2002. He's also the lead scientist in Brazil for the World Malaria Program, about which he'll tell us more about shortly. Dr. Moreira began his professional scientific training in the area of genetics and plant breeding. And as a postdoctoral researcher, began his study of the interactions of human pathogens and their insect vectors. In 2008, he began a 2-year visit to the laboratory of Dr. Scott O'Neill, who was then at the University of Queensland, Australia, who was actively involved in the study of Wolbachia and efforts to infect Aedes aegypti, which is normally Wolbachia-free, with a Wolbachia strain from the fruit fly Drosophila melanogaster, in the hopes of creating cytoplasmic incompatibility in this species, and as a means of potentially suppressing Aedes aegypti populations. Dr. Moreira made a remarkable discovery during that 2 years, and that was that the Wolbachia-infected Aedes aegypti that they had successfully produced were immune to infections by viruses such as dengue, yellow fever, and Zika. His publication in 2009 in the journal Cell is remarkable in its biology and its subsequent impact. This is a wonderful discovery and one that Dr. Moreira is helping to convert into a powerful public health tool. With that, let me welcome Luciano. How you doing? Thanks for your willingness to participate today. Glad that you're here. Let me invite you to go ahead. Let me invite you to go ahead and share your screen and tell me when you have it up, and I'll tell you whether I can see it or not.
Okay, just a second. Can you see it?
Great, I can see it. Go ahead and put it on presentation mode. Yes, very good, and I can hear you. And with that, okay. The floor is yours.
Thank you so much, David, for the invitation to be here today with you all here, and congratulate you with this initiative on this webinar series. I watched the last one, was wonderful. I learned a lot, and I hope to share with you today some aspects of the WMP, the World Mosquito Program in Brazil. So I will start talking about a little bit of the World Mosquito Program, which is present nowadays in 11 countries. As David said, it started in Australia and then went to other countries where dengue, Zika, chikungunya are a huge problem. So Vietnam, Indonesia, and other countries here. And then in 2012, we started the process in Brazil, and the first releases in Brazil happened in 2014. Later on, Colombia and Mexico also joined the program in Brazil. So the World Mosquito Program is a natural, all-sustainable, and not-for-profit initiative. In Brazil, it's conducted by Fiocruz, the Oswaldo Cruz Foundation, and involves the releases of Aedes aegypti with Wolbachia to the object of controlling dengue, Zika, and chikungunya. So David already gave a great presentation about Wolbachia, but I'm going to tell you more a bit. So Wolbachia is an intracellular microorganism, very, very common in nature. Around 60% of all insects, they have Wolbachia naturally. So the bacterium is transmitted through the eggs, so vertically transmitted by the females to all the eggs. And here I show the ovary of a mosquito, showing inside the oocytes here, in the cytoplasm. What you can see in green are all the particles of Wolbachia present here, and in blue are the nuclei. So as David said, although it's present in many insects and also other mosquito species, it was never found in nature in Aedes aegypti. So that was done in Scott O'Neill's lab in Australia with microinjection of Aedes aegypti eggs. Drosophila was purified from Wolbachia was purified from Drosophila melanogaster and then introduced into Aedes aegypti eggs. through this microinjection. In this process, it's very important to let you know that there was no genetic manipulation of either Wolbachia or the mosquito itself. So there were many scientific findings. David already mentioned this one, the cell, others showing for others arboviruses like Zika, chikungunya, and yellow fever. When Wolbachia is present in the aegypti, it can block the replication and the transmission of these viruses in these mosquitoes. So what's the main objective of the WMP is really to release mosquitoes with Wolbachia in the wild. So this graph here shows the wild population of mosquitoes, so you don't have Wolbachia in the beginning. And then we do a series of releases of both males and females mosquitoes. So it's important to say here what David already mentioned, the cytoplasmic incompatibility. When a male with Wolbachia mates with a mosquito without a female without Wolbachia, that female will become sterile. So that makes gives advantage for the females with Wolbachia to really to propagate and multiply and really to have more in the end. So we have this series of releases around 16 weeks, and then in the end we expect to have the majority of that local population with Wolbachia in the field. We can do both ways. We can do as adults, we can just walk in the streets and open it like a tube or a little plastic container and release these mosquitoes, or through a vehicle circulating the streets. And we can also use egg releases with these buckets or these China box containers like this one here was done in Australia. I'm going to talk about a little bit about the main steps here. So comms and community engagement, it's very, very important part of the program. So we start this around 4 to 6 months before we actually release mosquitoes. So we prepared lots of printed materials like posters, like these ones, pamphlets that explains the method for the population, and we distribute in many places. We also have these facts sheets for the health agents, because we work in partnership with the health agents in municipalities. So they explain how Wolbachia, what is Wolbachia, what's the program, and everything for the population to help us to engage that population. Especially now with the pandemic, we did lots of digital engagement. So lots of videos, lots of interaction in social media that was done over the past year with different actors. So the model for engagement is called Public Acceptance Model, PAM model. So it involves 3 main sectors. The health sector. So here are some photos showing the training and engagement of health agents in the particular municipality. We also identify and engage community leaders. So they are very important in this process, because they tell us how that particular territory will behave and how they are, what they do. So the best way to approach them. So this is very important, community leaders in this process. Also, we do lots of activities in schools. Of course, during the pandemic, in Brazil, the schools were closed, but those photos were activities that we've done in the past. So kids, and they're very good sponges to get the information and bring home and explain to their relatives, their parents, and explain about the program. And they're very curious about all the process. And then we also established a community reference group. In each municipality, we have this group. So this photo here shows when we did put together 2 reference groups, one from Rio de Janeiro and one from Niterói. So they got together after some time just to exchange information and experiences they had. So this community reference group is very important. So they are part of the community, different levels. We have teachers, we have lawyers, we have many different people that will come together. They meet twice every 2 months, more or less. And so we bring information and they give us feedback on how we are doing with the program. So they follow up the program since the beginning until the end of in a particular community. Then I go to production, releases, and monitoring. So other phases of the program. So this is just some photos of our biofactory in Rio de Janeiro at Fiocruz. Some photos of production of eggs and mosquitoes, adult mosquitoes, and then these tubes we use for mosquito releases. We produce around 3 million adult mosquitoes per week and around 3 to 4 million eggs per week, all of course with Wolbachia. And this is how we do the releases in many places. And so there's just a vehicle circulating, and we release around every 50 meters, around 150 mosquitoes, adult mosquitoes, males and females. In places where is lots of violence, because in areas in Rio they're very violent, and also places that we cannot get in, or because of the car is narrow streets, we work in partnership with health agents. So this is one of the health agents walking around with a bag and opening the tubes, and where the spots to release are mapped by our team. And then how do we do the monitoring? So how do we know if we are doing well, if Wolbachia is establishing in that particular neighborhood or area? So we install these BG traps, or we can also use OV traps. So we install these in people's and homes or commercial establishments, and then every week we go collect the little bag, bring to the lab to be screened, and then to send to the lab for quantitative PCR analysis. So for you, just to have an information, when we were in the peak activities in Rio and Niterói, we had 1,300 of these BGs installed. So you can imagine how big was the operation behind this to do all this monitoring every week we did since the beginning. So this is just a photo from the lab. So in our peak activities in Rio, also in Niterói, we had around 2,000 to 4,000 samples screened per week of mosquitoes collected in the field, just to give us the information how was the prevalence of Wolbachia in those areas. So I'm going to talk about a little bit from the beginning, the pilot studies in Rio and Niterói. So we had 2 pilot sites, very small, around 4,000 people only. So one is called Tubiacanga here in Rio de Janeiro, close to the international airport, and the other one called Ponto Final here in Jurujuba in Niterói. So 2 cities, they are linked by a bridge. So we did releases from August 2015 up to January 2016. So now in January, we completed 5 years since we stopped releases in these areas. Monitoring started in February 2016. Until today, we're still doing monitoring in these areas because they are the pilot sites. We want to know what's going on, but they are doing very well. I'm going to show you some results from Jurujuba, Niterói. So Jurujuba-Niterói is composed by these areas here in colored areas. They are kind of isolated one from another because of the geography. So we started 5 years ago here in Ponto Final, but then we went to all these areas here. So all the same neighborhood. In Jurujuba, we did not adult releases. We tried this egg release method. So just here in this scheme, we can see where we installed the we call the mosquito release container. So these little buckets here in people's homes, and then we put outside. And so after 10, 12, 15 days, mosquitoes come out. And we tested, of course, they are not breeding sites. They don't become a breeding sites because after this, we just go there and then replace the water and the eggs, mosquitoes with Wolbachia, food, and then larvae are produced inside, and then they come out. So this just to show you the Wolbachia establishment in this area. So here in these orange bars, you can see the number of samples collected over time since 1920 to 17, 18, 19. And then the Wolbachia infected samples and percentage. So you see here, year 1, year 2, year 3, after we finished the releases. And then you see Wolbachia here, just to have a normal common fluctuation here, but then became established and stable. And it's been like that over 5 years now. So we wanted to ask whether by Wolbachia being established after some time, what would happen with Wolbachia in those mosquitoes? Maybe it will decrease in density and number of bacteria inside the mosquito. So we want to test in 2 different times. We tested post-release times 2 to 4 months after, and also 14 to 16 months after the releases have finished. And we tested the Wolbachia density in these mosquitoes. So compared to mosquito gene as a reference, of course. So what you see here in gray are mosquitoes from the insectary, the ones that are being produced at that same time, and the ones that were collected in the field in green. So you see here, they have more Wolbachia compared to the ones from the lab. And then if you go back there and collect mosquitoes 14 to 16 months after, the same thing happened. And the whole thing is even more Wolbachia than the beginning of the establishment in the field. So it's good news. Wolbachia is still there, and it's more Wolbachia in those mosquitoes. We also, and then what we did, we collected eggs from these mosquitoes from the field. We brought to the lab, and then we challenged them with Zika virus and dengue virus, freshly isolated from the epidemics in Brazil. And what we saw here, these graphs show what? The Zika RNA copies in mosquitoes' head and thoraces from 2 areas. This first one in gray, Urca, are mosquitoes without Wolbachia, because there we haven't done any releases, so to serve as a control, compared to mosquitoes collect where Wolbachia was established. We did 2 experiments, so see the same results. Mosquitoes with Wolbachia, they have much less virus compared to the ones that don't have. And this is the same for dengue virus, mosquitoes with Wolbachia in green, much less virus compared to the other ones. Then just after the big epidemics of Zika, we got lots of attention in Brazil and also from our international funders. And we had this plan to really increase what we were doing as a pilot study. So the idea was to cover 946,000 people in Rio de Janeiro, all these colored areas here, and 373,000 people in Niterói, all this colored area here. So imagine we started these black spots here, and then we had to do all this. And of course, 20% of all these areas are, are like this. So in Rio and also in Niterói, we have areas that are very complicated, very violent, and our operations in these areas are much more complicated. Sometimes we had to stay 4 or 5 weeks without being able to enter there. Even the health agents, they cannot enter there because of the violence. We had to put our team into training because of for safety, and also how to deal in this in terms of when they see something, they have guns on, spotted to their faces. So all this happened during this time. So I'm gonna show you after the releases we've done. These results are for Niterói for March last year, because of the pandemic, we stopped the monitoring. All those BG traps were installed inside people's homes, and as I mentioned before, so we could not get in people's houses anymore. So we stopped the monitoring there. And what you see here, sorry, this slide is in Portuguese, but what you see here, when you see this green color, means that Wolbachia is above 60% in prevalence. In yellow, medium, 40 to 59, and in orange, less than 39%. So that's what we saw here. In Rio, in March, we had the island of the Covenant Island. We have areas that were above 60%, areas with 40 to 59, and these areas in orange, less than 39%. The main favelas are situated here, and we had to go back and try to implement. We're still doing some releases in these areas here at the moment. In terms of epidemiology, we've been doing some arboviral surveillance. This is done with our partners, the municipalities. So we get the data, we know when we did the releases. So we follow this up to see if there is any observational data that we can have some conclusions here. So I'm just gonna show some preprint materials that we submitted this year. So this is related to Niterói, and I'm gonna show you some graphs around that. So Niterói, we divided in 4 zones, and we also had this control zone. This control zone is important to say that was not chosen. It's just an area that's still missing to be covered. And in terms of epidemiology, it's similar to the other ones. When we had epidemics of dengue, Zika in the past, was more or less the same compared to the other ones. So here we started in zone 1, we went to zone 2, zone 3, and zone 4. This is in terms of chronology, when we started, and we went to here, here, and here, okay? So this table, just I want to point out the number of population covered in each area. So you see 23,000, 68,000, 178,000, 101,000. So it's around 373,000 people covered. And then the areas, just for your information about the size of the implementation. So here, show you in each area, zone 1, 2, 3, and 4, when we did the releases, and then the percentage of infected Aedes aegypti. So you see here aggregate percentage of Wolbachia in these areas here. So you see zone 1, it went up earlier because it was the first one, and then zone 2, zone 3, and zone 4 because we started very late by the end of 2019. So just to have we have an idea the prevalence of Wolbachia in these areas. In terms of dengue, I want to show dengue has a more longer historical data in the city. So since 2007, so maybe you cannot see very well all these graphs, but what you can show here, 2007, 2020, from zone 1, 2, 3, and 4, and this A, C, E, and G here. And B, D, E, H are the same zones, but is a shorter period, and the scale on the graph is smaller, so you can see more what's going on, because the last years, 2008, 2009, and 20, there was no lots of dengue and Zika in the city. So what you can see here in blue is where when we started following the epidemiology, following with the municipality in blue. So Wolbachia in light blue. In dark blue is Wolbachia here, so it's going up. And so Wolbachia here, Wolbachia here, and Wolbachia here. So, and then you see here the prevalence of the incidence of dengue in the control, in the dashed line, compared to the full line here from this zone 1. So you can see the difference. If you put in a large scale, you can see this difference here. And it's the same for all. So let's see this right side graphs. You see there is a difference in the intervention zone here compared to the control zone in all cases here. Zone 4, as I mentioned, you don't see much difference because it's much later in the process of releases. So what that does mean, that this data means, that we had a reduction from 44 to 77% less dengue happening here in these areas compared to areas in the control area, the areas that did not receive the program. For chikungunya was harder. They had the epidemic in Niterói. And so again, zone 1, zone 2, zone 3, zone 4, what the graph shows, the incidence of chikungunya, so the number of cases per 100,000 population. So you see here, control area compared to zone 1, you see a decrease in incidence of chikungunya when Wolbachia is established. So this is Wolbachia being established, Wolbachia here in dark blue and the shaded, and then you see in other areas. So that overall for chikungunya, we had a reduction of 60% when Wolbachia was present compared to the control area. So this is very similar to results that came last year in August. Indonesia, the WMP Indonesia finished a randomized control trial. So this is really the gold standard for epidemiologists to see. In our case, it's just observation data that we are following. So what they did, they had a city of around 312,000 people here that was in this intervention. So there was randomized. Some areas were chosen to selected by chance to have the intervention in light blue, and the white areas that did not receive the program. So they followed up hospitalized cases in this. In our case, the ones the data showed from Niterói is just a reported case in the municipality, but here the hospitalized cases that happened in Indonesia. So what the trial results were that they had 77% reduction in dengue incidence in these Wolbachia treated communities, which is great. That shows really as a gold standard method to show the efficacy. for Wolbachia being able to reduce dengue in this case. In Brazil, because of the results we had in Rio and Niterói, the Ministry of Health wants us to expand the program in other cities. So I showed you here in the Brazil map, we showed results from Rio and Niterói, which is just here. And then the next cities will be Belo Horizonte here in Minas Gerais state, Campo Grande here, and then you can see the population in each one, and also Petrolina in the northeast part of Brazil, which has 343,000 people. So the idea is we started last year during the pandemic with all the engagement in these cities. So the first one to receive was Belo Horizonte, and then Campo Grande started in December. Petrolina is going to start by May this year. So in Belo Horizonte, we are going to also have a randomized control trial, the RCT. The idea is to have 58 clusters. So this is already done. So this is the map of Belo Horizonte here. You can see here that these clusters in gray are the 58 clusters. 29 will be intervention and 29 will be control. So they were designed around public schools, elementary schools. So 3,500 children were recruited. The study is going to be funded by NIH, NID, and conducted by a professor here at the University of Minas Gerais in Brazil. We are going to only do the intervention based on this design. They will follow up with children that live in these areas to see the seroconversion for dengue, Zika, chikungunya. So they recruited these kids and they collect their blood, and they're going to follow up once a year until year 4 to see what happened after Wolbachia was established in 29 clusters. So I just give you some examples of the engagement that was done last year. So you see here some presential meetings that happened, everybody with masks, and we also did lots of Zoom calls to really engage, explain, train all the actors that would be involved. We are doing this really in partnership with the municipality, very close contact with them. So they are helping us a lot in all the operation. And for the RCT, we engage around 114,000 people, engaged by both WMP staff and also the municipality teams. So these were 430 activities with health agents, local leaderships, and also education professionals. And then we did really a pre-release survey. So this is a survey that is conducted in the city just to calibrate our actions, to see how is the knowledge of dengue and mosquito, and even if the people would understand a little bit about the WMP, if they heard something around. So this is not done by our team. So this was done by the health agents. And then I show you some results. Actually, the pre-release survey was done after all the engagement period. We had one which we call baseline survey as well, but this was the pre-release survey. So 95% of these 348 people that were surveyed said that they understood the Wolbachia method, and 87% agreed with mosquito releases. So 13% not. But if put this together, they understood and agreed, there's more than 90.6%. So this is very good. The majority of people said yes, just go ahead. it's very important that we don't want to force anyone. We just age, and so people are part of the program. It's not that we need to force to release mosquitoes in these areas. So the biofactory in Belo Horizonte is just some photos of the eggs, and then brushing the eggs off, and then the trays that we rear the larvae, and here the mosquitoes cages that we use to both to maintain the colony and also to produce mosquitoes for releases. A very important point here is that this was all refurbished, was an area that the municipality had, and it was for the first case at WMP Global that one municipality invested in their own facility. So all this production in one facility that was provided by the municipality. The staff that is working there is from our team, but they provided all the infrastructure. It's very important to remember, and everywhere, like when we do engagement, when we deal with the municipality, the health agents, that the Wolbachia method is complementary. We emphasize this in all our activities. So important to continue their practice at home to reduce mosquito breeding sites, to reduce, for instance, tires, garbage, and so on. If needed, and rationally to use insecticides also. If available, vaccines one day can be used in conjunction with the Wolbachia method. So very importantly, Wolbachia method is complementary. So with this, want to emphasize this. I just want to finish to acknowledging our funders and partners. We, Fundação Oswaldo Cruz, Fiocruz, with all the infrastructure. The Ministry of Health, since in the beginning, we have funding from them, and the Ministry of Health here, all these logos, and also from the Bill and Melinda Gates Foundation through Australia. Since the beginning, we are finishing up this year our last funding from them, and I think from now on, funding will be mostly from the Ministry of Health. We also had a little bit of funding for the epidemiology studies we've conducted in Niterói from CDC. And of course, I have to acknowledge our partners, which are the municipality health sectors and also education sectors in those municipalities. So because of the time, I don't want to take more of your time to show the activities in other cities, but maybe next time. Once again, I want to thank again a lot the invitation to be here with you, and I'll be ready to answer some questions if you have. I leave the contacts from our website and also digital social media here. So if you have some information, you can go and have information there. Thank you so much again. Thank you, David.
Great.
Thank you, Hector.
Good. Thanks a lot, Luciano. Really interesting program, needless to say, and the progress that's been made has been fantastic. So we have some questions that are coming in, and I encourage people that are in the audience to go ahead and ask your questions. I did want to start out by asking a question of my own, and just to get things started as well. There was, when you showed some of the data from your viral challenges of your Wolbachia infected field collected mosquitoes, there seemed to be some indications that there were some escapers of viral escapers. And I was wondering what you might be able to say about those viral isolates that seem to be escaping the Wolbachia immunization.
Very, very important, very good question, David. So I was a bit quick on that slide, but it's very important to show that in that particular data, what we are showing there was mosquito tissue the presence of the virus in mosquito tissues like heads and thoraces. So we just cut the mosquito, remove the abdomen, and then we process the rest, the head and thoraces. So you see a difference. Indeed, there are some head and thoraces with virus, but when we collected saliva from these mosquitoes and we see the transmission, it's mostly in our experiments we don't see virus being able to transmit. So the assay we do, we put mosquitoes salivate, and then we get the saliva, and then we inject in the thorax of naive mosquitoes, so, and to check if this saliva was infectious or not. And mostly we never saw a saliva that was able to infect other mosquitoes. So it's just indeed what I was showing there was just the mosquito tissue.
Yep, good, thanks. So I'm just going to start going down our questions here. The first one is actually two questions. One has to do with, let's see, can you release Wolbachia? I guess it's a question about the density of the Aedes population. Is there an efficiency in this method that relates to the density of the population? That's one question from this one of our participant. And the second question is really another technical question, and they were curious about how to separate pupae, how you're separating pupae and larvae from mass rearing.
Yeah, so the first question regarding to mosquito population density, it's indeed with what we saw. Firstly, many people ask that, do we need to do suppression first and then we come with mosquito Wolbachia? In the very beginning, the pilot sites, we did some just destruction of breeding sites just to have a natural suppression method there happening. But of course, going so big like this, we are not doing this anymore. What we do see that depending on certain areas where mosquito wild population is very high, it's harder for Wolbachia. It takes longer for Wolbachia mosquitoes to take over that population. So indeed, there is a bit of linkage there. And we are working on this to try to understand better what we are doing, or try to calibrate also the number of mosquitoes based on human population, because when you have more people, we have more mosquitoes. So we are working on this. In terms of separated pupae, we are doing a little bit with manual, with those glass plates for cages. We are doing a bit just to increase the number of females there. But for our colony, for releases, we are not separating. We just get, by the end of the larval cycle, we collect everything and we put in the tubes, and we wait a few days until the leftovers turn into adults. Even if the fifth instar, they will become pupae, and then we have some time, few days, to that pupae to become adults. So we don't separate.
Great, thanks. I guess I'm going to ask, there's a question from George Thomopoulos, who I guess relates to what I had asked to some extent. The question was, why do you think the field captured mosquitoes had higher loads of Wolbachia than the original lab-bred mosquitoes?
We are trying to understand this, and firstly, we thought that maybe because of the size of the mosquito could have an influence. But because we use a reference gene from the mosquito to do this density to compare, makes no sense. So this is not true, completely true. And so I don't know, maybe adaptation that over time, that was seen by another work done after a few years in Australia too, that happened, when they collect the mosquitoes from the wild that's been established, Wolbachia titer was higher than previously in the lab. I guess it's adaptation to the wild and to that mosquito that's been there already breeding and many generation of this interaction of Wolbachia and the mosquito. So it's really, I think it needs more work. I don't have all the answers, but just some hypotheses.
Mm-hmm. Yeah, okay. Our next question was, what was the rationale for choosing the additional release cities and sites?
Yeah, the rationale is, so we try to, in Brazil, there's lots of problems with arboviruses. We have lots of cities with problems, and you can't imagine how many cities contact us weekly asking us to come. Of course, there is an alignment between WMP Global, Fiocruz, and also the Ministry of Health to try to align what people want and also politically what they want and in terms of epidemiology. So what we try to do is really to get the most important ones. The rationale for these three ones that I showed, Belo Horizonte was a city that we've been working for a long time trying to implement. The city provided all the infrastructure, as I mentioned, but Campo Grande and Petrolina, the previous Minister for Health, he wanted to show that the program would work in different regions of Brazil, in different biomes of Brazil, like the Pantanal, close to Pantanal, where Campo Grande is, and also in the northeast part of Brazil, which is semi-arid area. It's very dry. So he wants to show. So next one maybe was mentioned, we don't have funding for this, but maybe go to Manaus, the Amazon, and see if it works there too. And so that was the idea. First, geographically in Brazil, to work in different areas, epidemiology, cities that have importance. And of course, there is some choices that we have to deal with that, like politicians come, I want to do in my city, and we have to deal with this. So all this happens.
Right. Okay, great. Adriana Castero asked a question about the persistence of the effects that you see in Wolbachia. Will continuous releases of WML infected mosquitoes be required to maintain the low viral transmission?
So if with the way we are doing now, so for instance, Niterói is a story that is almost the whole city. So what the Ministry of Health wants us to do is like a long-term monitoring, together with the epidemiology data that is coming every month from the municipality. So we want to follow up and see Wolbachia is there, is present. What's the epidemiology showing us? So that's the long-term monitoring of Wolbachia that's going to be done. Maybe every 6 months we are going to collect mosquitoes from those areas. But if everything goes well, as we are seeing already from the pilot sites, like 5 years that we don't do any releases there, Wolbachia is still close to 100%, sometimes 100% and weekly basis. We are doing okay. So if Wolbachia persists, I think we'll be fine. And with this long-term monitoring and epidemiology following up from the municipality will be important too.
Yeah, okay, great. Thanks. I got a question from Nikki Faber, who says, first of all, really cool work and a great talk. Thank you. And I have a few questions. So I'm going to give you a couple of them. How do people generally react when you tell them there's no genetic modification involved? Do you think this might give genetic editing a bad reputation when you sell the Wolbachia mosquitoes like this?
Yeah, so this is very important question. Indeed, when we started, there was some misunderstanding that we were working with transgenic mosquitoes, and people reacted saying, most of them, if it's transgenic, we don't want. So of course there is we need to tell, explain, and this difference I think makes a difference. I don't know. So if we bring, of course, there is the Oxitec in Brazil. Everybody saw last week's presentation. Great presentation. Great work they are doing here. But I think it needs to be better explained. People have to be aware of it and be sure that they understand that's the transgenic. Of course, for Oxitec, there is no, it's not sustainable. It's going to be there forever. It needs work, it needs engagement, not forcing people to accept the methodology.
Yeah, I guess if I could just interject a question here, is to sort of riff onto that a little bit, does the issue of the fact that this is an infection that isn't found in nature in this particular species ever come up, or is it just sort of, you know, it's not something that comes up and
it did sometimes, yes, but when they understand, like for instance, I think the fear is like sometimes like people raise the fear that okay, Wolbachia was not there, was put in the aegypti, and then it's going to be going to many others. And so if we explain like they share breeding sites with other mosquito species that have Wolbachia already, and that transfer did not happen over time, and it's Wolbachia is already present in other mosquitoes that bite you. You have Culex for such a long time in Brazil, and it's been bitten by Culex mosquitoes that are positive for Wolbachia and also Aedes albopictus. So I think if you give the information, people are more they accept and they understand, and then they become a partner.
Yep. Let me just get to a second question that Nikki Faber had, does Wolbachia help against malaria also?
Yeah, so there are some papers showing. So firstly was just injection of course Wolbachia in the mosquito, and then it did have impact on Plasmodium. And later on was able, group Xiongzi was able to have a line of Anopheles stephensi with Wolbachia. He showed also that was blocking Plasmodium. And then later on, everybody thought that there was no Anopheles species naturally infected. And there were some groups in Africa found some particular places where some mosquitoes were Anopheles mosquitoes with Wolbachia naturally. But when they try to bring to the lab and study this, the maternal transmission is not 100%, so it's much lower. And also the CI sometimes not present, the cytoplasmic incompatibility, which makes difficult for field implementation. But it has impact on Plasmodium, yes.
Yeah, great. I'm going to give you one more question from him, from Nikki Faber, and it has to do with the overall goal and whether you think it's possible to fix Wolbachia in the global mosquito population. Sort of the range in which you imagine this taking effect.
Yeah, as I said, for instance, if we bring things to Brazil, everybody asks, oh, how long is it going to take for Brazil to have Wolbachia everywhere? So Brazil is huge, and of course we have problems in not in Brazil, but many countries in America and Asia, and that have lots of these problems. I think really, as I mentioned, Wolbachia to be used as an integrated control method and with other things not only this, we are not the silver bullet. We need to be in conjunction with the education programs, people doing their jobs, and so the municipalities, and also insecticides if needed, vaccines, and all together, I think will make a difference. It's not saying here we should release mosquitoes with Wolbachia everywhere. That's going to be the solution. That's my point of view.
Mm-hmm. Good. Well, let me keep encouraging our participants or audience to ask questions if they have any. Here's one from Michael Eckersoffer, who asks, what's causing the post-release fluctuations of percentages of Wolbachia infected mosquitoes?
Yeah, great question. I think it's really the sampling. Sometimes it happens. For instance, we get a BG trap and we have one, like 2 mosquitoes. One is positive, one is negative. So you have one with Wolbachia will be 50%. And if you have 2 or if you have more, so it's really sampling. This has this fluctuation. It's more. It's worse when we have less mosquitoes captured in our monitoring system.
Yep. Okay. Another question from Adriana Crostero. It has to do with will viruses develop resistance to the effect of Wolbachia?
So this is one question that we have a lot, and in terms of evolution, of course, it's possible. I think it's going to take long time to this to happen because the mechanisms are multiple. It's not just one. There are at least 5 that's been published, like different things that are happening inside the cell that Wolbachia is already there and really is blocking virus replication and the completion of that cycle inside the cell to be able to continue. So because it's multiple mechanisms, I think that evolution I will take longer. But if it takes, let's say, 30, 40 years, we already will have a great impact on the epidemics of dengue and Zika and chikungunya, for instance.
Yeah. Do you think you could expand a little bit more on what the mechanisms, some of the ideas about what the mechanisms of resistance are? You mentioned that there are multiple ones. Could you just say something about those?
Yeah, there are a few studies showing, for instance, immunity, like when Wolbachia is present, you have more like TOL, IMD, that pathways are upregulated, and these will produce molecules that will block the virus there inside the cell. In terms of competition for cholesterol, for instance, the Wolbachia, both Wolbachia and virus use. So just a few to mention. And then, of course, when Wolbachia is there too, there is some others proteins that are changed, that are needed by the virus to replicate and then to have the envelope and be able to finish its cycle and go out of the cell to be able to. And it's very important to point out that not every single Wolbachia strain that blocks viruses, and just a few. It's not known why too. But it's also important to show that where Wolbachia is present in terms of tissue, like in the midgut and also the salivary glands, tissues that are important for the virus to replicate and then to leave the mosquito body when it bites another person. So this is also where it's located.
Yeah. So in order to get this immunity effect, is it only cells that are infected with Wolbachia that are going to be resistant to viral infection, or is there a systemic effect in the mosquito that, or is it completely cell autonomous?
Yeah. So I think David, I think there are need, although many papers came out on this, the mechanisms and also these pathways that all, I think there is still need for studies to come and really to understand better what's going on. So we thought it was just a cell and decided that particular cell, but it might be more than that, as you said. Maybe it might be cell autonomous, but I don't have information at the moment to give you like proof for that.
Yeah. Here's a question from Angela Meyer, one of our in the audience. How do you guys handle the operational difficulties of refreshing water in mosquito release buckets or servicing monitoring traps during large events like Carnival?
Yeah, that's a very good question too. Yeah, indeed. Sometimes it happens that we have to stop that particular week and do nothing because it would be a waste of time. For instance, as we work in partnership with the health agents, and if there's the holidays, they will not be around. So we have to really to plan in advance. And of course, even planning, there are some changes to accommodate what we need to do and also their reality, their routine in the municipality. So this is also, I think, is also important to say when you talk about implementation and talk about transgenic mosquitoes. If we had this questioning, like one thing that happened a lot was the health agents being worried that they would, let's say, if this works, I will lose my job, so I will have nothing to do. So this is more on another layer that we have to work and try to talk to them and say, of course, if you have less of these, you can work on these other problems that you're already doing. So the municipality can just shift the focus on other diseases and other things that are needed.
Yeah.
I just want to include that.
Yeah. Last week, our webinar, as you know, was given by two folks from Oxitec, and they described their new delivery system that they're using, which is an egg-based delivery system, and they described these boxes that they would distribute, and then that seemed to be, from listening to them, that was and thinking about it, it really was somewhat of a game changer in terms of how they can distribute their product. And both through the mail and also stockpiling it because of the ability to store desiccated eggs for a certain amount of time. Do you think that and you mentioned that you did in fact use some egg releases in one of your sites? Do you think that that modality has potential for you guys?
Yes. I don't know if Scott O'Neill is here because he would love your question, David, because he's always, he says that I'm stubborn in Brazil to still be using adult releases, which it's, it seems to be a pain like to do all this operation. So what we are doing at the moment, just to say other countries in WMP, other countries, they are all doing egg releases, and it works very well. We showed in Niterói, but it takes more time in the field for, if you want really to do like a quality control or quality assurance, we need more people to go inspect those buckets and see if everything is well. But we are doing a pilot now in a favela in Rio, starting now in February, which I think is going to be very nice. Was a project from our team. So it's just a gaming system. So we are going to give some phases for this game that the community will participate. So we will have distribution centers, the health units in that particular favela. They are going to have the egg buckets there, the egg, everything, all the materials they need. So they bring home and they have all the information, they have videos to see how they do to implement. So, and then you have ways to show, to send photos of what's going on, to follow, and then they come back and they give scores. And so all this gaining in the process of using the community to do the implementation. So that's a pilot we are doing. And of course, if successful, I'm sure that we're going to expand this to other areas in Brazil.
Great. So I think we've come to the end of our questions. I don't see any others there. I'm going to, maybe I can just wrap it up then, Luciano. Let me say thank you very much for presenting today and telling us about this fantastic project that's really taken off as a result of that remarkable finding that you made a number of years ago. It really is quite exciting, and the epidemiological effects that you're seeing are really significant and quite unique. So again, thanks a lot. Really appreciate it.
Thank you, David. Thank you for the invitation again. Thank you for all the participants here.
Good. And just before people leave, I just wanted to remind them that this webinar series will continue next week. We have one scheduled entitled "The Development of the SIT package against Aedes mosquitoes, Progress and Challenges." And this will be given by two experts from the FAO IAEA program in Austria, where they're developing SIT technologies for and other things for mosquito control. So with that, let me say thank you to the audience. Thanks again to Luciano. Thanks to the FNIH events team for supporting us. And thank you, Hector, for being my co-host today. So with that, we'll see everybody next week.
Thank you very much.